How's it going, everyone? It's Chow here, and today we are going to talk a little bit about lipids. So let's get started. So lipids are very fascinating. They're actually one of my favorite topics to discuss. But what's really interesting about lipids, unlike many of the other types of macromolecules that we talked about, for instance, carbohydrates or proteins, is that lipids are technically not true polymers, as in you don't really have monomers that kind of build up to make a polymer. So if you think back to a protein, you have monomeric units, the amino acids, that build together to form this big old chain of protein. Same thing goes for carbohydrates. You have these monomers called monosaccharides that you build together to form your oligo and polysaccharides. But lipids don't really follow that exact type of pattern, so oftentimes we don't really consider lipids having monomers, and that lipids really don't have sort of polymers as well. Obviously, depending on who you talk to, that might be debated, but so is the case in a lot of science, right? So with lipids, they're generally nonpolar hydrocarbons. So you can see a bunch of nonpolar hydrocarbons back here, the fatty acid tails, and they're often the big old group of individual lipid components, for instance, triglycerides or phospholipids. They're often at least connected and held together a little bit by these weak forces called van der Waals forces. Normally, they're fairly weak forces, but when they add up, they can hold fairly tightly. So when it comes to lipids themselves, they actually have a good number of functions, just like proteins. Perhaps not as much as proteins, but there are some key functions. So the first one is energy storage. You probably know that lipids can be used as energy storage, especially longer-term energy storage. There are also structural components as well. So for instance, phospholipids can act as structural components for your cellular membranes, which are obviously very important to the survival of yourself as an individual. And then we have steroids and other modified fatty acids, for instance, via hormones or vitamins. And these are very key too because they can help regulate the body in certain ways. For instance, steroids can bind to DNA and actually affect transcription rates, for instance. And we also have thermal regulation and thermal insulation. That's obviously very important. If you have a lot of fat on you and you're standing outside in the cold weather, you feel nice and warm. So thermal insulation is very key. And then obviously we also have electrical insulation as well. So if you think to, for instance, with a neuron, you have myelin sheaths around those nerves that can actually potentially increase the depolarization and the traveling of the signal and the speed of that signal, and it's often very important for the function of a particular organism and getting some kind of detection or feedback within that organism from a certain stimulus. And then obviously with lipids, they repel water, and so they can be useful through certain actions like swimming or floating. So if you have an animal that has a lot of fat in them and it's fairly aquatic, it can allow them to stay afloat, etc., etc. So when it comes to lipids, one of the key things that we have to talk about is the fatty acid. So the fatty acid is this yellowish group back here with these two tails. These are the fatty acid tails. The fatty acids are very fascinating because they are really just chains of nonpolar hydrocarbons. So it's just a big old chain of carbons and hydrogens. What's really interesting about these fatty acids is that on one end there's a carboxyl group. So over here, that's your carboxyl group. And then on the other end is a methyl group. So there's your methyl group over there. And these fatty acids can be broken down into kind of two main categories. The first one being saturated. So the saturated fatty acids are the ones without any sort of double bonds between the carbons. And then you have the non-saturated fatty acids, which do indeed contain double bonds in that carbon-carbon chain. And obviously this can affect the biochemistry of a certain, for instance, cellular membrane. And depending on what particular fatty acid it is, it can have one double bond, so it's monounsaturated, or it can have multiple double bonds, which make it polyunsaturated. So we're going to take a look at some of these examples right now. when it comes to saturated and unsaturated fatty acids, it can actually have a huge impact on certain cellular membranes, certain globs of fats, and seeing whether it actually stays liquid at a certain temperature or solid at a certain temperature. So if you look at in this instance over here, you can see that this is a saturated fatty acid, so there's no double bonds between the various carbons. So this is palmitic acid, and you can see that they're all single bonds between carbons. So again, you have your carboxy side over here and then your methyl group over here. And if you look at non-saturated fatty acids, so they have double bonds in them, you can see that there are also variation as well. So this over here is one particular type of fatty acid. This is, I believe, oleate. And you can see that oleate has one single double bond, which makes it kinked in that location. So this little bend over here in this cis formation is actually a kink, and so there's one single double bond. And this particular molecule here on the right, which is linoleate, you can see that it's more or less polyunsaturated, which means it has more than one double bond. So this and this is going to generally be more fluid at room temperature than something like this. And this all has to do with stacking instances and how the chains are able to interact with one another and stack together, and therefore it can affect whether it stays solid or liquid. So keep in mind saturated versus unsaturated fatty acids. Now, we can also talk a little bit about triglycerides, which is this really fun thing where you have a glycerol head over here, and then you have 3 fatty acid shales. So what's really fascinating is that you can actually make triglycerides using those fatty acids to form the fatty acid tails and the glycerol. So through another dehydration synthesis reaction or condensation reaction, you lose water and you end up forming ester linkages. So you form these ester linkages between the glycerol molecule and the fatty acid molecule, and you can make a triglyceride. So a triglyceride has 1, 2, 3 fatty acid tails, hence triglyceride. Now, I'm sure most of you know at this point, but if you have a huge buildup of triglycerides, it kind of acts as adipose tissue. That's where it ends up being stored, and that's what we call fat. And depending on triglycerides and many different types of fatty acids, you can make various combinations of each. So you can actually have triglycerides that are a little bit varied in their fatty acid chains. So if you have a lot of saturated fatty acid chains, which means that you have a lot of carbon-carbon single bonds, and then you have very few, or I guess I should say in saturated fatty acids, there aren't any double bonds between carbons, you actually allow closer packing. So it stacks much more easily, and therefore, because of that easier stacking, they're solid at room temperature. Whereas in other instances, if you have unsaturated fatty acids for that triglyceride, and this is the case for honestly not just triglycerides, but any type of lipid that has fatty acid tails, but if you have an increase in the amount of unsaturated fatty acids, you can actually cause more kinks in the chain. So this is just a kink. It's very kinky, as they call it. And when you have a kink that happens in an unsaturated fatty acid and you attach that to a larger molecule, such as a triglyceride, it doesn't pack as tightly. And because it doesn't pack as tightly, it's more likely to stay liquid at room temperature. So if you have a high instance of unsaturated fatty acid in a certain molecule, you end up with an instance of that particular molecule being more likely to be liquid at room temperature, or at least it stays liquid across a lot of the lower temperatures. Whereas if you have something that's like this with a lot of saturated fatty acids, you're going to have it more be solid. So with animal fat, for instance, there's a lot of saturated fatty acid, hence the reason why they're solid at room temperature. Whereas if you have plant oils, oftentimes it's going to have a lot of unsaturated fatty acids, and that's the reason why they stay more or less liquid at room temperature. So there you go. Now, when it comes to fatty acids and lipids, there's one very key point that we always like to discuss, and that's the phospholipid. So phospholipids are very important parts of our cellular membrane. But what's really fascinating about phospholipids is that they're similar to triglycerides, but instead of having 3 fatty acids, they only have 2. So they only have 2 fatty acids that are still bound to this glycerol molecule, but instead, in the third position, there's a phosphate group replacing the third fatty acid over here. These guys are amphipathic, so they're a molecule that has both hydrophilic up here and hydrophobic chains, which makes them both polar and nonpolar. So polar up here, nonpolar over here. Hydrophilic up here, hydrophobic down here. So you break this molecule into kind of 2 different sections where you have a hydrophilic head with the phosphate group up here, and then you have a hydrophobic tail, and that's your fatty acid tails. So phospholipids are amphipathic, and they have both polar and nonpolar groups. Now, when it comes to phospholipids, again, it's a very key component of cellular membranes, and through natural orientation, you actually can have the hydrophobic tails that actually point towards one another, and then the hydrophilic heads that point away towards the solvent, towards the polar ends, towards the water molecules generally, and that allows for a good orientation. And of course, it can bend over, so you can have this bilayer structure that comes back to the other way, forming what's sometimes known as a liposome. So you have an arrangement due to the hydrophobic interactions between the fatty acid tails, and the tails actually the tails end up kind of aggregating towards the center away from the water, and through just natural orientation as well as a little bit of entropy, you can actually create a buildup of something like this. So the hydrophilic heads point out and in, and then of course the hydrophobic tails are pointed towards one another. So you have a zone out here that's going to be full of water, and you have a zone over here that's full of water, and this area over here is going to be devoid more or less of water. So it forms a natural structure like this via a bilayer, and this is sometimes called a liposome. So there we go. So what's also really fascinating is this idea of steroids. So steroids are basically hormones that can sometimes diffuse into the nucleus and affect rates of transcription. And what's really fascinating about steroids is that oftentimes steroids are going to be made from cholesterol. So cholesterol here acts almost as a precursor for pretty much every single steroid out there, and it shows this unique kind of tetracyclic ring form. So you have 1, 2, 3, 4, and these are the 4 tetracyclic rings. And if you see a tetracyclic ring formation like this, oftentimes it's going to be some kind of a steroid, or perhaps a cholesterol precursor to a certain steroid. So there you go. Now, last thing that I really want to discuss is this idea of vitamins and waxes. There's not too much information, generally speaking, that you have to know about them for an introductory biology course, but vitamins are generally molecules that are not synthesized in the body, or they are synthesized, but they're not synthesized in large enough sufficient quantities, so they must be acquired through the diet. Hence the reason why sometimes we call them essential vitamins. So they have to be eaten through the diet in order for you to obtain those vitamins, which are also very important for the body. As for waxes, really they're just highly nonpolar and impermeable to water, and they're a type of lipid. So I'm pretty sure we've all seen a candle, and we kind of know that, hey, that's a wax. And what's really also fascinating is that that in itself too is a type of lipid. So here is just kind of a summary of the common lipids that you're likely going to see in an introductory biology course, as well as maybe a biochemistry course. So again, we have cholesterol over here with that tetracyclic ring structure over here, and it's a precursor to, again, steroid hormones. So if you see something with a tetracyclic ring formation, it likely is some kind of a steroid or a cholesterol precursor. You also have a free fatty acid over here without the glycerol head. And you can see over here in this particular individual that this one has one single double bond, so it's mono-unsaturated. And then with this one is a triglyceride, so you have the glycerol molecule over here, and then you have 1, 2, 3 fatty acid tails. 2 of the fatty acid tails are basically saturated completely with hydrogens, whereas there's 1 over here that's unsaturated with a single carbon-carbon double bond. And then last but not least, very important, a phospholipid. These are the things, again, that make up your cellular membranes. And you can see that in this particular individual, it's a little interesting because it has the glycerol group over here, you have the fatty acid tails, and then you also have the phosphate. And what's really also fascinating is that you can actually have additions to that phosphate group to make it different. And you can have additions to those via the phosphate group, as is the case over here. So that's basically it for lipids. I hope you all found this at least semi-helpful, and I will see you in the next video. And as always, best of luck studying.